Yes, 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 please, please. कोणी माझ्या रिमोट एखादा मोबाईल हा फोन થી જોઈએ ને બટ એ મહેનત કોણ કરે બધું બરાબર તમને બરોબર હો આપણે પછી કઈ તો ઘટે નહીં ભાઈ જો કરંટ છે Yes, so good morning to all. After a long time, 2 days is a very long time, right? Really difficult to come to college even for you, even me. Sometimes Monday is a very difficult day for everyone to restart our education field, but we have to. So I think you have completed first lecture, so you are now recharged. I hope it is recharged. Okay, so let's see. Let's move ahead. So yesterday, few days back, just I was talking about the reproductions. Mainly thing is what asexual, sexual, or the other modes. We are going to learn about certain things. What's going to happen? That the DNA mainly a transfer. One cell has acquired the DNA, whether it is in case of animal system or in a plant system or in a microbes or fungi, anywhere, them. It is going to acquire something new. What the viruses are they doing? They are just duplicating themselves. So one form of a cells has acquired or distributed themselves into new. This is just making a copy and paste. Xerox machine is going on. Everyone is looking for their survival. What is the survival here in a means? That once a species has established, age will come, then they are going to die. So what they are looking, that they are looking to keep their lineage to be get survive. As we are, our father, forefather, grandfather, and it goes on, we are on a certain stage. Population is increasing. Okay, but even after the government is having certain regulations in certain countries, then after that also lifted. Earlier it was okay. So earlier it was been lifted. It's like a China. Only one child policy was there. Now they have lifted. The reason is what? Or any rewan. કામ આવે છે પાછળ આવે છે ચકેડીમાં બેસવું હોય બરાબર અને ચેરે રહેવા દો આપણે આમ જ બેહો આપણે મૂવમેન્ટ કરવાની જરૂર નથી. So what was that policies that's going on? So like this only, every microbial population even looking to survive. So they are growing at certain stage. They are looking. It's a leg to log phase. They will grow. One within 24 hours, they will reach a stationary phase. Maybe in 48 hours. You can play with the microbes in different modes. We are going to perform some growth curve. If you initially give like 100 ml flask is given or 250 ml flask is there, you have filled with 100 ml nutrient broth. We are going to inoculate about 0.1 or the 1 ml bacteria we are going to add into it. And we have kept into incubations. So it will start to take the nutrition and they will grow. In other words, what you can do, sometimes it is like you may, while preparing what you are doing, the end broths, some material is written like 2.8 gram or something is there. Then the nutrition availability is more. The bacterial cell will move in the log phase more, leg to log. But if it is if you going into 1/4 of their concentration. Suppose you want to play with the microbes. Okay. So what going to happen? It is like you are in a class, right? Your capacity is 4 lectures a day or 3 lectures a day. If I want to make a something, okay, I may repeat I'm going to call the another people's also, and 8 hours a day lectures may go. Okay, it will be a 12 hours. What going to happen? You are going to be a lethargic. You are attending but not going to attend anything, right? Same like that. And if the energy wise, if I'm looking every day 4 hours, I may give like in a business or something like that, or in a workshop somewhere. It is per day some meal is given. I may give the meal just in a 1/4 quantity. What's going to happen? You will feel that we are hungry. Okay, you are hungry, but the material is not provided. 4 lectures you have to complete at least. So what you are going to happen? You feel lethargic, energy will going to deplete, and you cannot move. You have to be in a system, but you are attending but not that much attentive because of energy depletion. Same thing going to happen in this or bacteria. If you are going to initially, when you are preparing the nutrient broth, suppose you are adding 2.8 gram. Okay, what is the 1/4 part of the 2.8? 2.8 gram is a suppose 100%. If I'm looking for 1/4 part, how much amount will be there? 1/4 part of a 2.8. How much it is? 0.7. Yeah, it will be a no need to have the calculator. Suppose one can calculate even 7.7. So initially 100 ml flask you have taken, you may just add the 0.7 ml gram of a nutrient powder you may weigh, and you are going to inoculate. Now there will be a fighting. You have added 1 ml, fighting will be there. So bacteria will come in a lag to log phase. But nutrition depletion will be there. They firstly going to move in a stationary phase. So we are going to play with the microbes in that way. So what sort of the thing is happening that nutrients availability is the major thing in always in every case. So there is a sexual reproduction, asexual. Bacteria generally following into binary fission. Here our main targeted part is what to understand about them is a binary fission. So they are not following the mitosis. Generally our cell cycle. Here the basic chart is that we are going to learn step by step more that bacteria is going to double their material, genetic material. This is called the DNA replications. First is the R phase, or you can say the replicative stage. It will be followed by the division. So there will be a division is going to happen and the two cells will generate. And here to once back to go into cycle, it's called the interphase. Same it is we have learned supposed to be in our cell cycle for womb in the eukaryotes. How the cell cycles always run, it is like a whole is the interface. We know that like G1, S and G2. What's happened in the S phase is the DNA replicative stage. Before that, cell is start to grow. Here it is written as a cell growth. I hope you understand about the cell growth and development. Right. So growth, they have started to gather the material. They are getting mature to get divided. So DNA replications again, before going to mitosis, the cells will start to collect the material to divide into two. It's like a property to be get divided between the two, their siblings. Maybe it's a girl or boy. Okay, sometimes only 2 boys are there. Or it is, if you are gender neutral, if you are talking between 2 siblings. So equally one has to be given. So they will gather, the cell will gather every sort of things, and the cell is going into phases. Karyokinesis followed by the cytokinesis. So this is happening into what? Eukaryotes. But what about the prokaryotes? Prokaryote does not having this capacity because there is a no-nucleus, right? There is a no-nucleus, then there is a no specific mechanism, and bacterial chromosome is single one. There is a no many number of the chromosomes are there. Single one giant chromosome is there. Yeah, along with it, it do have some plasmids that we are considering as a what? Extra chromosomal DNA. So bacteria will, during the DNA replications, will double their all the material available to them. Sometimes the number of the plasmids changes. The same plasmid, maybe in a copy number. Here it's the word called the copy number. The plasmid is the same. Maybe it's the 10, 15, 20 is there. Why it is maybe a copy number change? There is a extra thing will be loaded over it. It is like antigen resistance, antibiotic resistance. Maybe in a nearby area, some microbes or some pathogens are grown, and they does not allow growth of the others. So what they will do, they will synthesize more. It is like arms. When you are going outside, you know that this area is very problematic. So you will keep every sort of things in bags and everything. If required, I can fight or shoot something. It's depend upon the area. If you are going into certain places, otherwise you are very much vigilant. Anytime anything can happen. So bacteria will be vigilant by adding the plasmids, their arms. So they will increase the number. If you are knowing that if I'm going with only one arm, maybe it is taken by someone, so I can going to use some other arms. Okay. Ultimately, lastly, two heads is there, legs and others part. That is for the fighting. SOS response may come, but before that, you may have some arms located. So this arm is going to get used. So maybe it is like comparative. Can be asked that the difference between like prokaryotic and eukaryotic cell cycle. Okay, so don't get confused that I've never been heard. So cell cycle you have learned about it, right? Maybe in earlier in the past somewhere. For the eukaryotes, so we are going to learn about the prokaryotes in a way that how they are doing. So generally, there is no specific specialization. Here there is no mitosis. Okay, mitosis is just a place, it is not there. So what's going to happen here? Here you can see from the daughter cell to here you can see like a mother bacterium. We are going to say similar. It is like a cell division is going to happen. So mother to daughter cells, right? So here you can see that is a one formation called mesosome. On the membrane, one protuberance will going to form. It's called mesosome, where a chromosomal DNA is going to get attached. First place into it, where our bacterial chromosome is going to get attached. After the attachment, it's going to duplicate it. So it is called the replicative phase. DNA replication is going to happen. At the attachment place, it is going to get duplicated. As the cell has get duplications, it is going to acquire the material and the cell plate is going to form. As it is get duplicated, cell elongation. Bacterial cell will going to gather. It is like a DNA replication is happen, then the G2 phase. In case of previous, here it was DNA replication and the G2. Here it is a G1. So here you can write from daughter cell to G1 phase is there. From the G1 phase, the bacteria has started to develop their DNA. So it is a replicative stage. It will be followed by the cell elongation. So it is in a similar to the G2 phase. Now it's not going in a mitosis, but similar process to the mitosis will be carried out. So what's going to happen? It is going to form a range of FTZ proteins. Here the one group of proteins are participating. It's going to prepare a line FTSZ. Many a time in so many examinations I have seen in objective, they are asking about this question. Name of the protein that participating in bacterial fissions. It's a group of protein participating into cell cycle, bacterial cell cycle. So don't get confused. Cell cycle is there or not. So they are going in a cell cycle. So here it was going to start and develop a membrane-like structure. You can say the membrane-like structure is going to form, and then a septum is going to form. Septum is nothing but a protuberance. Membrane will start to develop afterwards, and it's going to continue after separate formations from the both the end. From both the ends, septum is going to form. It's like a bubble. If you are here playing in the going in kindergarten, right? So many kind of it they were having. From the one itself, you can round and make a two, but it cannot be separated. If you are going to cut from it, it will blast, boom, and two cells going to not form here, but here it will be. Right, here it will be going to divide. So it is like a very easy. It's like you are having the one sandwich. Okay, sandwich is there. Or when you are playing in a vada pav, bread was cut between to it. You can just open up, you can separate into two. The size is going to reduce. In vada pav, forget about the pav, vada, just a pav. Okay, so what you are doing, you are first initially using the some knife to cut it. Cutting down, and you are going to separate upper part and lower. So original cell is divided into two. The size has reduced. You can see here. Size is equally half distributed. So it is like a cell cycle in a bacteria. Many a time this question can be added in 4 to 8 marks. People are asking. Here your pattern is something different. 4, 6, 8 is there. So it can be asked. And the major important questions will related to ring or the membrane-like structure is going to form. It is FTSL. Whenever I'm going in many exams, here it related to here or somewhere else, it is been asked. So don't forget it. So very easy. What are the phases are there? It is equivalent to following the G1, S, G2 and M phase. But we are not going to write it. Okay. Here we are going to write what? I phase. Here I have just written in the next point somewhere also.
Most bacteria.
Yeah, binary R phase, D phase, and I phase. So bacterial cell. Bare જાઓ તમે. મોઢામાં આંગળી નાખીને મનાય છે. ક્લાસમાં please move out. You don't know, that is why I'm telling. Many a time I have told to everyone, I am not going to allow anybody in a lab or in a class putting something in mouth, cutting their nails, everything. You don't know, that is means you are since long not present mentally in my class. You may move out. You may move out of the class. Thank you very much. You have called earlier to me, right? Regarding the practical. I don't know what you are putting in your mouth. Yeah, just doing. Yes, just doing it. You are accepting it or not? By submission of yourself, you are accepting that I was not cutting the nail, but I'm putting my fingers in my mouth. Thank you. You are allowed to come in the next class. No need to again ask that I'm not allowed. Thank you. So here we are not going to write M phase. Most important part, if you are just, while questions can be asked, if you have written the M phase, is mitotic phase. So it is not. Here we are going to use the word division. Division phase is there. So from where it will start? Here. Cell elongation to till it's divide. As the cells is there, daughter to mother cell, it is called interphase. Interphase means cell is static. Whether it is in lag phase to log phase or stationary phase, it's going to wait. That should I go for the cell division or not? If it is a log phase, it will continue. They are just dividing, divide and increase their population. If it is stationary phase or in death phase, they will wait. And one has to wait, otherwise the growth will be there. Okay, growth and development we are looking, they should go in the developmental stage. Physiological maturities need to attain. So we are going to learn about some next points are there. Batch culture, continuous culture, synchronous culture, something. So sometimes we are looking that our bacteria should start to develop, sometimes the alcohol or certain production is required during the fermentation. If they are just dividing, so they are not going to develop any sort of the metabolites. So they have to be attained in the static condition. So in a way that the cell may move into our stationary phase. So we are going to plan accordingly. Our practical sessions may be developed in a way. So just one small documentary I have kept.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
So first step is what? Mesosome formation. Pardon? Be louder. I am not able. Mesosome. In this, they haven't shown about the mesosome. Okay, that's what I'm telling, that they haven't shown mesosome. In the some documentaries you can find, you may go in YouTube, so many animations may be there. You can just find it out. Okay, so first step is what? Develop the mesosome. So where our chromosome, bacterial chromosome is going to get attached, then the replications will start. It's going to duplicate their bacterial chromosome, and then a septum formation. Before the septum formation, one membrane-like structure. It is the FTS said proteins is going to develop one membrane. So it is the place from where it's going to get divided. And it is get matured, the septum will form from the both the end. Once it is get matured or the septum is ready to develop, it is going to divide into two. So mother cells is going to equally divide into two, and the size is going to equally distributed. You can say if it is the volume maybe a 1, both going to get the a 10.5.5 volume. So what we are going to say here, the phases we are looking, it is the interface. Daughter to mother cells going to be in interface, then the replicative stage, DNA replication, and followed by the divisional phase. Here you can write about G1, G2, but it is not. Okay, it's maybe equivalent to it, but here the terminology is somewhat different. So there is a R followed by the interface. It will be followed by the R, and the last is a D phase. Clear? So same it is written. I'm going to I just forgot to upload such things in your Google Classroom. Within 1 or 2 days, I'm going to upload this PowerPoint, so you can also read it out. Okay. Yes, so it may take, generally, this cell cycle running in E. coli, it has been understood, or practically they have come to know, it's taken 20 minutes. So last time I have asked you people to do a calculations, right? 1,000 organism, what will the number of the microbes within hours, then in a next day. So within 3 to 4 hours, they will become millions to going in a millions to billion. When you are looking on a next day, you will find the billions of the cells. So just I have asked the number like 1,000, 10,000, 10 lakhs or more than 10 lakhs. So everyone has came with certain calculations that I will going to discuss in the growth curve. How we can calculate very easily the number of the cell populations in our culture. So that will discuss. Before that, anybody having any questions in this cell cycle in bacteria? It can be asked in a some different manner also, prokaryotic cell cycle. So here we have taken the example of E. coli. Okay, measure all the bacteria following this format only. It is going to remain same. It is the mitosis we are telling. It is not like that in animals and humans. Mitosis is going to remain same. Okay, same it is in a bacteria. All the bacterial cells are going to follow such pattern. So anybody having questions, doubts? Be louder somewhat. No, there is no much difference. Here it is a protuberance is going to happen, right? So is a membrane and cell wall is going to form along. Here it is a pushing. It is going to follow the protein. Here the FTS protein membrane. It will be followed here. So here the whole cell wall and cell membrane is start to introduce. Once it is attained at the one place, it is going to get separated. So it is following the protein. So here there is a no different protein is not septum formation. Okay. Anybody else? I hope it's clear. Okay, fine. Then we shall move to the. So I have divided all the topics into some of the point wise, so it's become easier for us to understand. Yeah. So next point we are looking for the sexual reproduction. Okay. So into it what? Here it can be certain advantage and disadvantages. What could be the advantage into it? That is genetic variety development, genetic variation. When the bacteria is just dividing and dividing, okay, there is a chances. It is like an Asiatic lion. It is a very great pouch. It is in Junagadh. Giru area is there. So when just 2 to 3 years, about 3 years back, one virus has infected their lions, and about 20 to 30 lions get died. Then after directly the dosage, such research was not carried here. In South Africa, there is a one different line is there. South African line is there. So there, the researchers do have the certain vaccines for it. It was not for the same virus, but it was a different. So that vaccination or their medicines were given to this Asiatic lion, and they survived. So when any pouch is very less diversity is there, is the more chances of going to get the vanished of that species or the genetic race. It is like that you are here from the so many different places are there. Okay. Some of the material or the some bacteria infections may cause to everyone, some at a time. Or something is given in a food, which was a poisonous. But everyone is going to have the different response because they're already from the indigenously, they do have the resistance. If it is from the how the hygienic and unhygienic conditions from the childhood you have followed. Those who are in very hygienic conditions, sometimes from the Australia or from the US people are coming, they have to take so many medicines and the vaccinations. Otherwise, on a first or second day, they are going to fall ill. One is the diarrhea, and the second, it is like a fever. But if you are going here to there, we feel not such. Like a Asiatic group or the African groups are meeting one to another. So there is a maybe a chances. So from their childhood, some of the one or another way they have exposed certain environment, so they can survive. So if there is a genetic variation is there, then whole species in the strain is not going to lost. It was like a corona has came. So many persons got died. Okay, if by chance some again the some potential viruses may come, maybe half of populations get died. Some may survive. So genetic variations only and only possible with the sexual reproductions. Why? During the mitotic to meiotic division, okay, we know that during this time crossing over is taking place into prophase 1. Have you learned the meiosis? Somewhat, maybe. Okay, so I'm not going to recall otherwise, no problem. So there is a, you can say the recombinations may take place and the genetic variations is takes. So every year, even in the same family, a child from the same biological parents may be not 100% same. Siblings also differ. Okay, so siblings may also differ. Yes, twins. There are the congenital twins are there. If their genetic DNA is also similar, in that case, there is a called the microenvironment. There is a twins are of a two, maybe two eggs were formed and fertilized by two different sperm. Then the twins may develop, their genetic makeup is different. Congenital is there, from the same embryo, they have divided into two and they independently they have developed. So genetic makeup is same, totally. But even after they are growing, they may differ. The reason is what? Then the playing the role by the microenvironment. What is meant by the microenvironment? How they have developed, in which environment they have developed. It is like a someone has seen the like movies like Parvaris, very old in the Bollywood. I'm telling Amitabh Bachchan, Vinod Khanna, right? In that time, earlier movies like that only. Their childhood, that they have changed their child. Okay, someone is abducted or it is happened. So when a villain is there, they are going to have the child from the good parents. Good parents has thrown to there. Okay, but then after they may grow in different formats. So development is very much important. So where it is developing, it's called the role of micro environment. So even a twins is going to happen, but the chances susceptibility of their twins, it may be higher. That one is going to get the some disease, another maybe. It is like a sometimes we are calling parents do have like diabetes or certain disease conditions are there. There is a chances of getting on their child because of genetic material. Okay, but in that conditions, people already know, and they are getting married with some other guys who is having the very much resistance into it. Then the less chances of their child. So sexual reproduction is going to give the variety of genetic. Yeah, what could be the disadvantage? More complex, slower because of must find the compatible partner. In case of bacteria, even every cell is not going into this kind of a sexual reproduction. It may take a time to find that partners. Okay, and no new individuals produce. So there is a no increase in population at higher level. Here there is a transfer of material. So we are going to learn one by one. Here called the conjugation breach. Okay, E. coli do have the certain pili-like structure. Generally, it is being used into what? For a locomotion. But the same pili can be used for the development of a sexual breach. Here you can see they have developed the sexual breach. So what's going to happen in this case? They do have, there is a special kind of F plus. It's called the fertility plasmid. It only having the capacity to develop the pilus. All the cells does not possess it. Every cell cannot going for the sexual conjugation. It's also called the conjugation. Conjugation cannot be formed. So it's conjugation breach. Conjugation breach is formed when it having the fertility plasmid or F plasmid. Once it is developed, what's going to happen? It's going to replicate it. It's called the F elements. Here it is known as the F elements. So this F elements is going to duplicate it and transfer to their partner. Okay. It is called the rolling circle DNA replicative mode. Maybe you have learned about the DNA replications, right? So when it is a plasmid, in the bacteria also, they are following the rolling circle model. It is like leading and lagging strand develop in the DNA replications. Okay. Fork-like structure is formed and the DNA get replicated. Right. That part we have semi-conservative mode of DNA replications. But here it is following the rolling circle. So just opening and the development of this. So between the which conjugation breach, it will transfer their genetic material. And with this, sometimes what is here it is called the F plus cell. This one is the F minus cell. Earlier it was F minus. Now it's became F plus. F plus means what? It do have the fertility plasmid. Here you observe in case of a bacteria, there is a no population is developed. It is those sexual reproduction. Okay, those sexual reproduction, partner 1, partner 2 got attached. Once again, they are separated. After the separation, they remain as such. Number of the population is not increased. Yes, in case of eukaryotes, it will be there from the mother, not from the father. From the mother, there is a development of a child is there. There the population is increasing. So difference between sexual reproduction in prokaryotes and eukaryotes. It's the measure is what? Here the no population increment, while in case of eukaryotes, the population is increased. Getting understood? The reason is what? There is a no much difference. But yes, reproduction is called why? DNA is transferred. One important phenomena for the reproduction is what? Every new cells is going to get something new. And here it is new gifted by this. So gifting is done by what? F plus cell. It's going to develop first a conjugation bridge. From the conjugation bridge, the rolling circle model, plasmid is transferred to the next partner and they are going to get F plus cell, all the fertility elements. What is the advantage of it? Many a time it has been observed, this F elements, initial part is remains same. Okay, it is required for the development of conjugation bridge and to transfer. The remaining part, it do possess so many genes. Maybe it is antibiotic resistance. Okay, one important part. As well as for some bacterial cells do have the in a plasmid genes related to enzyme or related to some degradation. It may be a petroleum product is there or any unhygienic for them also. For us, unhygienic is what? The bacteria. For them is also nearby another bacteria or certain chemicals like a pollutant is there. So by chance, any pollutants may come. They are floating. Okay, you have so many bacteria. You have just washed. So via washing, they have gone in sewage. During the sewage, they were happy with us, but you have lived them. So they have now gone in a sewage. So if they do possess resistance material, then they can survive. So via F plasmid transfer, they will survive very easily. So there is a mode of transfer in case of sexual reproduction is just related to what? Plasmid transfer, DNA transfer, and the population is going to remain same. So question can be asked in a way that the difference between sexual reproduction in prokaryotes and eukaryotes. Okay, so in case of prokaryotes, if it is also like pollen, or in case of animals, there is a sperm and egg cells are there. Okay, they are going to, in haploid conditions, going to bound with one to another. They become diploid, and the zygote formation is happening. And once again, they became a whole individual. And the population is getting double. But here the doubling is not. Getting this concept? Major important point is what? No population growth. Now this cell both will going into binary fission, not a mitotic. They are going in, they are following that normal protocol. Binary fission is a normal protocol for them. Only in certain conditions, they develop this bridge. Right, just previously what I have shown, yeah. Bridge development. And it's also not a usual. It's not a real that much usual to be get developed. In specific conditions only, they are developing certain bridge. So F plus cell is ready. Okay, they are ready with it, but only in certain conditions when the proximity of the 1-2 bacterial cell is coming nearby, and they are here, there is a less chances of development of the conjugation bridge between F plus to F plus. Okay, so they, when they are growing in certain area, in a normal binary fission, the populations all of the F plus cell will be there. So they have to find their partners who is F minus. Okay, so when you are growing certain cell in your flask, all are the F plus, then there is a no pilus formation between them because already both are the same. Even if yeah, sometimes in certain conditions it's been observed that the pilus formation is happens between two F plus cell even, but there is a no chances of the getting movement because their membrane structures will already knowing it is there. Or by chance if one is start to, if the switch on and switch off is there, then the first who initiate rolling circle model to transfer their genetic material, they can transfer to the F plus cell of their partner. Another will be get inhibited because already in a bridge, it is like a one way, it's moving. If you are in the 6th floor and 1st floor, okay, from where the lift will start, it will move. Okay, so if it is like the first to initiate, So maybe you are on a 6th floor. From the 6th floor, lift has started. So DNA will transfer to the first floor to there. So F plus, very rare chances. But by chance this may happen, then the only one is able to initiate their mechanism. As it is, the fertility material is transferred, the pili formation is breakage. Pili is going to break and they individually going to be separated into 2 different cells. So during their pili formations, generally between the F plus to F plus, very rare chances. By chance this may happen, only one partner is able to transfer their material. Okay, so such phenomena is there. So they have to find F minus cell to their nearby proximity. And this proximity is coming in the micrometer size. The size development is also very less. It cannot develop here to that last. It will be a very lesser. Okay, and this proximity is coming in occasional. And that time only they are able to develop. So I hope it is clear till this. Once at a time cannot DNA translations, the replications here. Yes, it's going to be a copy of this. Once it is transferred, here we are referring with what? F plasmid only. Fertility plasmid is called, F element, so many terminologies are there. So it became replicated. So both are now separated. So it is going to remain similar to one to another. Both if it is F plus by chance. Suppose here the conjugations between the 2 F plus may happen. Then one partner is only able to transfer the DNA. Both together not change. Both together cannot proceed.
Because there is no condition of new cells. Can you call the bacterial conjugation reproduction?
Yes, we are learning as a sexual reproduction only. Sexual bridge has formed. That is why we are calling it as a sexual reproduction. Important phenomena is what? In case of reproduction, DNA transfer is the only meaning. DNA transfer is the only meaning calling as a sexual reproduction. Third, we are going to look once again different stages. There is a transduction we are going to learn. Transformations we are going to learn again. Okay, so from the environment we are going to learn into next. Okay, just let me show it. Okay, it's in the next PowerPoint, no problem. Yeah, so it is other mode of a DNA transfer. So here I will take this conjugate transductions later. Let me talk about few minutes related to transformation. Yeah. Yes. Another mode. There are the other mode of a reproductions we are going to learn too. Transduction and transformation. Transductions is taken place by the bacteriophages. Okay, and transformation is taken by 2 different ways, with DNA fragments or with the plasmids. So sometimes in a population, when it is in stationary phase, cell has died. Okay, they are not looking to divide in binary fission, and cell has died. So when they died, sometimes their nucleotide division is fragmented. Sometimes they are get divided because of a sometimes shearing. They burst even, or the cell died. And in this medium, some DNA fragments are lying. Okay, these DNA fragments, it's going to take uptaken by the some bacterial cell. It may going to get degraded or sometimes going to get integrated within the bacterial chromosome. So Here also we are going to say as other mode of reproduction. DNA uptake from the environment has taken place. Even the animal cell has that tendency. Even a plant cell, when plant cell without the cell wall, we are terminologize them as a protoplast. It has the tendency to uptake. It is like a DNA vaccine. Have you heard about it? That people are looking for the DNA vaccines, means directly going to inject into the muscles. So what is the vaccine purpose? They are going to develop certain protein. If this certain virus is there, so virus do have their protein code. For the protein code, we are going to recognize that only in a vaccine. What is the main motto? To identify a material. Okay. So when it is like it has been told someone is roaming, by chance, if any a person with a green t-shirt or the orange t-shirt need to cut, nothing else, security guard only knowing that part. By chance, someone has came out from the outer side. He is wearing the green t-shirt, but he is not from the Marwadi, but it has been caught by security guard. Okay, he's not looking for the logo. Okay, he's in green t-shirt. Let me cut him. Okay, it's like a vaccine. We are going to introduce a protein or the DNA directly in DNA is injected. So muscle cell is going to develop a protein. Protein is moving outer side on a membrane or in outer side. Our army of the WBC will find them, and they will develop their instrumental things. They may go for the human immune response, or you can say cell-mediated immune response may arise, and it's going to get arrested and cleared. Like that only. Or all the cells, all the individuals, even it is like our mentality. Something is freely available, we may take it, right? If we want to pay something, we may not. But if it really is given, let's see. If it is useful for me, I will address through. If you are moving outer side in a class, sometimes they are giving some material, freely material in a mall or something. You may take. You are interested, you will follow, otherwise no. Or so many times it is been given. Sometimes it is like a party, right? Or some of the places there, you will take everything in a dish, right? Let me just test. If I will like, I will go for again. So this is what a tendency to all the organism to take everything as much as possible from the outer place. Like that only cells will do. They will uptake. So it is not a asexual reproduction, not a sexual reproduction. We are going to call them as a other mode. It is really difficult to terminologize something here. So other mode of reproduction, and there we have termed new as a transformation. So many times what we are doing, that transformation is done artificially. What we can do, either in a plant cell or animal cell or in a bacteria also, we may doing the electroporation. Means you are taking the some cells in a vial. Okay, give the jerk, electric current. What's going to happen if you are going to put your finger into electric power? Current will follow. Sometimes your hands even going to get a blackish, right? So cells, when a microvolt jerk is given, pore formations may happen. Their cell membrane going to get the pores. Okay, once the pores is formed, you have added from the outer side DNA fragments in append of tube. When it is done, you have inserted with electrodes, and it is done. People are doing the electroporation method for preparing the genetically modified organism. So what's going to happen? DNA or either plasmid. Sometimes whole plasmid is also taken up. So you have already added the plasmid within the sample. Okay, and you have given the jar, it's going to take. While some of the time it's remain open. Pore formation is there. During that time, transfer of the material from the cell sometimes losing, they are taking something. Okay. So whenever you are in a lobby, okay, or something may happen, earthquake or jerk has came. Everyone is trying to move from the class. Some students may came out, some will move in a class or in a canteen or somewhere, or trying to move to down. So it is like for them, it's earthquake. So anyway, material is going to transfer here and there. And there is a chances of getting it. So it is called transformation. So it is other mode of reproduction. A new terminology has came as a transformation. So clear with this concept? 5 minutes left. I'm not going to continue. Yeah, then after it's going to recombinant. Sometimes, it may, if it is a similar, then there is a chances of material transfer, or it may integrate as a part. And during the evolution, if you are looking, we are knowing that functional and non-functional, so many genes are there. So it is acquired during the evolution. It has just acquired a DNA from their environment, whether it is necessary or unnecessary. It just has acquired, and the genome size increased in so many numbers. Functional genes are very less. Coding and non-coding DNA, if you are looking, okay, junk DNA sometimes we are calling. So we are having the lot number of the portion as a junk DNA. It is just copy, right? It is like that you are having when you are going in a tour or somewhere you are going. In a luggage, we are taking enormous of number of the things, right? Water bottles and this for the beautifications and so many things and so many extra clothes and everything we are taking. Sorry, we are not going to use everything, but we are taking. Like that only they have acquired. Yes, ma'am. Yes, that's just I'm simply telling. There is called the recombinance. Recombinations either may happens or by chance they are going to get integrated if the recombinant material, if the same matching is happens. Okay, same matching is there, it is going to replace one to another, or else they are going to get integrated. Okay, because it's a genetic material is lying, sometimes it may remain in a round circle form, in a form of a plasmid. Okay, so it may integrate because genetic material here in bacteria are looking to acquire something and something from outer side. It is like your class is there. Okay, someone is coming, you will say, let allow him to sit. You will never mind. 10 students are coming, you will never mind. Okay, 50 students are coming, you will never mind. After one time, you will say, what is this? We are not able to survive or search. So then it will not integrate. Limitation is there. Certain periods, some of the materials get accepted, it's going to get integrated. There is like a nick formation. I'm not going to take you in more molecular details right now. There is a very specific recombinance frequency is taking place. Very specific mechanism is also there. Some are REC proteins. I think I have taken, yeah, here it is. I have taken into the next slide. Okay, so what happened? Bacterial chromosome transformating DNA. There is a free nucleotides. REC-A protein is participating into this process. Recombinant A, that is called the REC-A protein. Detailed mechanism we are looking to interested into it is called the REC-A protein. Every individual cell sometimes does not possess this REC-A. If they do possess the REC-A, they will able to get it. Okay, so REC-A protein is participating into it for the integration. So these cells should be competent enough. In evolutionary stage, if they have developed the REC-A, then they can. It is like you have got so many materials. What you are having, some pockets and wallets. You can put something. So you have kept certain place, but afterward your pockets are full. Okay, sometimes without pockets, if you are moving, right? Then where you can hold something in your hand, into hands like this, but afterwards you will not. So it is REC-A protein. Without REC-A, also so many other mechanisms are there, but we are currently not looking to learn about it. So many mechanisms are there. Molecular biology is full of wonder, full of wonders. Every individual cell do have during the evolution has developed so many new technologies by themselves. Okay, so currently I'm not taking, but the REC-A is participating into transformation. Okay, so sometimes may happen the free nuclease does not allow double-stranded DNA to be get inserted. It will allow only a single strand. So this single stranded is going to bind with REC-A. It's going to find the place where it get integrated. Okay, when it is going to get integrated, it will going to insert it over there. So there is a homology. Homology means similarity of that material is there, then it may transfer. It is like the someone is coming, is going to substitute someone. Okay, one person is coming, that means class remains strength same. So one is ready. Okay, I was just waiting, someone can come, I can move. Okay, so they will allow to go. In another format, if this is not there, no similarity, one is going to sit with one to another. It is called the process of transformation. Other mode of reproduction. Even here, no population increment after recombination, after transformation. See, there is a no doubling. Yes, now this cell is ready for binary fission. On next step. So they only able to increase in population by asexual reproduction only. No any other mode. Clear up to till now? Everyone? I hope. If it is not there, in next class also, you can ask. Not always. Sometimes some material is coming, it is useless. Resistance may came. Here this copy has came, which is of a no use sometimes. But the size of chromosome has increased. Someone is coming in your class. Okay, he is remain idle. He is not even performing nothing, but he is now become part of your class. But yes, someone has came. Every time, questions, many things. Topper in a class, you will find that, oh my God, someone has came, having the signing activities, or maybe a notorious. So, or the idle guys also are there. So chromosome size has increased here. It may giving the some important phenomena sometimes, sometimes not. It is chance phenomenon only. Understood? So let me put the links for your attendance. Those who remain to put their attendance, they can do.